So welcome to our third video in this demo part of the course G08 on metagenomics applied to viral genomes. We're looking at functional analysis, or really starting in that direction. What we're going to use is a database called Logan, which is many contigs of nearly everything in the NCBI SRA. So this is a very large data set, 96% of the SRA with 27 million sequences that comprises over 50 petabytes of data, or as compressed context would be 384 terabytes. It's a really large database and it's cloud-based, which means we can search it ourselves, and that's what we're going to do with our context. And you see here the extent to which it is different types of information in kind of the orange color versus NCBI in black. You can see there's a lot more information in many taxa when you look right across the tree of life. So what we're going to go to is this website on ORIS. We're going to search, well, not even one contig at a time. We're going to use a text search just for a fragment of a contig. It will split this contig into k-mer length of 31. We can adjust the stringency threshold to be higher or lower, and that reflects the number of k-mers in a sample to be considered a match. And that will take our query to query them against we're mainly interested in contigs that can be matched. So we can use Google Chrome to do this in Oryx, or just directly copy and paste it over like I do to the Chrome browser on your laptop. The maximum length is 1000 bases, and you'll have to submit the the query as a text query and load it on the website. You'll need to add your email address, and you can toggle the groups criteria to be all just till we get a better comprehension of what might be present. Is that these take a few minutes to run, and ideally you should take at least one of your viral contigs and one of your metagenome contigs to see what you get. So what we expect to get is a table of accessions. So you get your ID, the k-mer coverage, the bioproject, biosample, bioproject ID, a globe map showing the sample distribution across the world, a frequency plot of the match types, and the last match details if of interest. So you can see if we look at our adenovirus genome, this is what we get. So we can see it's only located in one particular region, and it's this level of matches. You can see this is the table of the matching unitags based on sequence length of one, and the matching positions that correspond to this particular SRA library. This is for metagenome node 1. You can see that it has more matches, and these are quite different. They're a soil and run of metagenomes, so they're quite not really the same, and they're for different accessions as well. And you see if we look at the distribution of matches for the metagenomic sample compared to the viral sample is just one match to the adenoviruses, where the metagenome matches many diverse environments. And you see that if we look at the spectrum environments from which those matches come from, you see it's quite a big mix, and reflecting obviously where the sampling has been done across the world as well. So that kind of brings us to the end of the practical, and we'll check that Logan search on the server shortly. But one thing you can think about is your approaches for A and B, which is better and why, what the strengths and weaknesses of the different approaches. Bearing in mind that for output A, we're looking at Kraken2 that gives us taxonomic information. We can extract the Kraken2 to use a taxonomic identifier to get the viral contigs. We can de novo disassemble those contigs, and use checkv to look at the quality, and also use Logan search to explore that as well to confirm our match. We can also just directly de novo assemble to get the mag, and use Logan to explore it. It's just one of many databases that are available, but it's newer, so it's worth trying in addition to the ones you might know of already. And then we can use checkv to explore that as well. So I hope you see they can kind of actually be complementary approaches. So let's then look at a server to see how we can run Logan search on there. So we can, for example, take our virus sample and get the contigs for that, which are here. And all we need to do is take a chunk of the first node, because that will exceed the limits on Logan search if we go beyond this. So we can go to the dashboard. You can paste it in as a text query here. Hit load to load that. Give it your email address here. And then for the groups, give it all. And then you can maybe increase the threshold if you don't want to do any matches and submit that there. So you can see the website is Logan search. And then we can do that as well for our virus, our metagenomic sample. So we're going to extract that as well in the same way, just using conjug 1 initially. And we're just going to use that as a text query again. You can use more advanced approaches that are obviously not involving a website. However, they're cumbersome and won't fit into the link that we have available to achieve that. So this is merely to just give you an idea of what you could do with that. And it's up to you to assess if it's useful for you or not. So let's submit that as a job. I see that's running, and that will be sent to your email address if in case you you have a very long query or if something else goes amiss. So these are now running. What we could even do is look at the other ones in the out sample, which are quite long. I can't really show it on the screen if I do it a different way. Let's look at conjug 2 just to see what happens. So this is the other conjug for node 1. So we can take that and again go to the website. Load the query. Make sure the groups are all at our email address. Increase the threshold perhaps, and submit that as a job. And these will all run and hopefully complete shortly. So when we look at the viral context that we get for the adenovirus matches, we can see that we have one single match to this particular SRI ID, which corresponds to a bioproject associated with phylogenomic analysis into these adenoviruses. We see the prevalence map here showing just one location, and that includes the GPS coordinates as well, or the longitude, latitude. You see there's just one single match here for bottlenose dolphin adenovirus 1. And then if we do look at the blastoid alignments, we can look at the match that we have and search to retrieve those K-mer matches. But they correspond to the different reads that are inside that particular library. If we look at our metagenome sample, you see that there's many diverse matches, most of which are associated with soil and runoff metagenomes from different locations. You can see if we look at a map across the world, that's quite broad and reflecting sampling locations. If we look at the plot, you can see there's a lot of diversity there, most of which the strongest match is just wastewater metagenome and soil metagenome and plastic metagenome. And then if we look at blastoid alignment, we'll get a similar sort of match. If we look at conjug level search, and we do that per match, so it's quite a long list when you think about it here. If we look at the second conjug, again, you see more wastewater metagenomes that appear here. Again, they have a broad geographic distribution that's slightly different. Again, you see wastewater metagenome for that one, and the blastoid alignment will give us the same sort of thing. So you can see how Logan can tell you more, and this is in addition to the other data in the other databases that are widely known, as well as the typical pathway level analysis that you could do. It's just a different perspective to harness the abundance of information that's available. And so you could take all these identifiers and do further analysis using information from those outputs as well. So please look at the intro videos and the course instructions for how you can extend your learning and the resources that are available There's many other metagenomics courses that are available, so please look at those as well.